Hello everyone, my name is Ivan de la Rubia and I'm going to talk about our new tool for the reference-free reconstruction and error correction of transcriptomes from Nanopore long-read sequencing. So first of all, a brief introduction. Nanopore sequencing is a new sequencing technology where the molecules pass through a pore, producing an electric signal on each base pass, as you can see in the animation. And then this electric signal is converted to the actual bases using what are called base colors. So with nanopore sequencing, we can sequence DNA, cDNA, and direct RNA. And in the case of DNA, it produces ultra-long reads up to hundreds of thousands of kilobases. And in the case of cDNA and direct RNA, it can produce full-length reads. And it is also much more portable and cheaper than Illumina sequencing, so you can plug it into a laptop almost anywhere. But it has several disadvantages. One of them is that the error rate is still really high. In our case, where we use direct RNA and cDNA for building transcriptomes, the error rate is comparable in both scenarios, and it averages around 6% of insertions and deletions and around 5% of substitutions per aligned read base. So we wanted to build a tool to work with cDNA and direct RNA in order to cluster the reads that originate from the same transcript together, then perform an error correction to improve the error rate, and finally, with this information, produce a final transcriptome along with the expression level of each transcript. And we wanted to do all of this without using a reference genome or transcriptome. So there are many reasons why it is a good idea to make this without using a reference genome or transcriptome. And some of them is that some diseases express novel transcripts that are not in the annotation, or some species don't have a reference genome or transcriptome, or is not of quality. So we built RATTLE. RATTLE starts with a set of long reads from the sequencing step, and then it performs a first clustering step where reads are clustered together at the gene level. So reads that originate from the same gene would be placed together in the same cluster. This is done using a two-step greedy algorithm so that it performs reasonably fast, and there is more information on the algorithm and other benchmarks in the preprint. Then the gene clusters are separated into transcript clusters, and once we have these transcript clusters, an error correction step is performed. The reads are aligned using a multiple sequence alignment algorithm, and then they are corrected using the consensus from each column and taking into account the quality of each base. For example, in this column, 3 reads have a gap and 2 reads have probably what is an insertion, so the insertion is removed from the actual read. And finally, an extra step of clustering and error correction is performed, and the final transcriptome is output along with the quantification of each transcript. And I'm going to show a few results. So we've benchmarked our tool at 3 different levels: at the clustering level, at the error correction level, and then at the transcript quantification level. Here we can see the adjusted brand index, which is a metric to see how good the clustering is for 3 different clustering tools. In this case, we've used simulated data with Deep Simulator, which is a tool to simulate nanopore reads based on a deep learning method. And we can see that RATTLE outperforms the other 2 tools in this scenario. We've also benchmarked the clustering using real cDNA and RNA data, and we can see that the results are even better in this case compared to the other 2 tools. Regarding the error correction, we can see how RATTLE improves the percentage of identity, being really close to 1, and the error rate is really close to 0 compared to the raw data, which is the pink box. There is this other tool, TranscriptClean, that offers better results in terms of identity and error correction, but it is a reference-based method. And finally, we've checked the quantification using self-spikings, and we can see that the correlation between the predicted abundances, which is the y-axis, and the actual abundances of the spikings in the x-axis is actually pretty high. RATTLE is the only reference-free method in this plot, and it gives correlations really comparable to those that are reference-based. And to end the presentation, I'd like to thank all the team that is working on this project in Barcelona and in Australia. And you can already have a look at the preprint, which is in bioRxiv. In the preprint there are many more details on how the tool works, there are many more benchmarks, and you can also try out our tool in our GitHub. And thank you for listening.